with our plenary on the policy landscape in the United States. My name is Michelle Mello. I'm a professor of medicine and law at Stanford, and it's my pleasure to host the panel today, which includes some very experienced commentators from both government and research institutions. Key findings 2 and 3 of the NSABB report address the adequacy of the policy frameworks in the United States. And the second finding indicates that the frameworks overall are effective, yet finding 3 suggests that their adequacy for managing the risks associated with gain-of-function research of concern may vary depending on which pathogen we're talking about. So there seems to be plenty to discuss on the panel today about where the policy frameworks may and may not be adequate or optimal for addressing these risks. To that end, we've asked our speakers to reflect, depending on their institutions, on the issues facing federal agencies in administering this regulatory framework, as well as some of the strengths and weaknesses in our current policy framework and opportunities for optimizing our oversight of this area of research. So the speakers' bios are in your packet. I won't belabor introductions, but we'll just start simply with Dr. Jerry Epstein, who is Deputy Assistant Secretary for Chemical, Biological, Radiological, and Nuclear Policy at the Department of Homeland Security. Thank you.
Thank you very much, Michelle, and thanks to the academies for giving me the opportunity to speak with you today. I am the ex officio member of the NSABB from the Department of Homeland Security, and I've been working closely with the working group as it's developed the proposals that we're here to discuss today. It's not the role of an ex officio member to tell an advisory board what to advise. That's why we have outside advisors. So I'm not going to be providing opinions on what I or what DHS thinks of what they've given us, but it is our job to try and provide technical and policy support and help make sure that whatever recommendations they do provide are in a form that will be most useful to the U.S. government when it takes this up and then has to come up with an ultimate policy. I've been asked today to help describe the existing policy landscape that affects gain-of-function research. So I'm going to talk about a variety of proposals, some of which I think are very familiar here, some of which are less so, in terms of the context within which life science research is already being done today. And I'll try to touch on those areas in which it overlaps with the recommendations made by the working group. And first, so let me just very briefly summarize that. We've had a very good discussion of that already. In terms of the working group's proposed gain-of-function policies, I have it characterized by a number of different questions. So first, who's covered by it? Directly, it affects research institutions that receive U.S. government funding. In terms of which pathogens are covered by the proposal, as has been noted earlier, it's not a list of pathogens, but rather a set of criteria. Experiments which are reasonably anticipated to produce a pathogen meeting the criteria on transmission, virulence, and spread in human populations would be captured by the NSAB proposal. So again, not a specific list of agents. And in this respect, it's unlike the funding moratorium we now have in place, which is agent-specific. Which activities are to be governed by the proposal are, again, those reasonably anticipated to generate a pathogen with those problems. That was not Freudian, I hope, with those criteria. And what does the proposal require of research proposals which are found to be in that set? Is that there are 7 principles must be satisfied before funding is to be released, and then once it goes through the standard funding process and is evaluated compared to other possible funding claimants, the research would then undergo appropriate federal and institutional oversight policies. So in a nutshell, I hope that has not mischaracterized what the working group has recommended. So in terms of the policy landscape, there is no existing set of policies that addresses this particular set of research. That's indeed why we've asked the NSABB to come up with a proposal. This, as characterized here, this gain of function of concern, is not something that in total is overseen by existing policies. But there are a number of things in place which touch on bits and pieces. So let me go over a bit of them now. If I can have the next slide. Oh, I've even got the clicker. In terms of existing policy context, I want to differentiate between those which are in effect by force of law and therefore affect all research within the United States, as opposed to those which are, say, a condition of government funding, which would directly affect only the recipients of that funding. And of course, there are indirect effects, as has been mentioned earlier, in other areas. But directly, a funding hook would only affect recipients of U.S. funding. Those parts of the existing context that affect all of us by law are ones I've selected here. First of all, are the statutes in place to prohibit biological weapons development. This prohibits work with biological agents for use as a weapon or in types and quantities that are not reasonably justified for legitimate purpose. Nothing specifically about this statute says anything about gain-of-function research. And presumably, if that research is being done for legitimate, bona fide research purposes, which is an actual explicit provision that's permitted in this law, it would not be affected by this. But it does provide the context and the ability of the federal government, should there be egregious acts of use, the development of a biological agent as a weapon, this would permit prosecution of that. This law is also the mechanism by which the United States implements the Biological Weapons Convention, an international treaty which prohibits development or acquisition of biological weapons. That is not just a treaty, but it's a violation of U.S. law by virtue of this provision. Now, this provision does not have anything specific about types of activities or agents that would be deemed use as a weapon. It's a very subjective determination, and for that reason, would become very difficult to prosecute someone, even if there were activity that seemed hard to justify. And partly for that reason, the select agent regulations were developed in response to a second statute. This is a comprehensive set of safety and security requirements governing any use of certain listed pathogens. And here, you have to be registered, you have to be vetted by the government, the institution has to have permission to use those agents. There's requirements on the safety and security and incident response having to do with use of these pathogens. And here, one doesn't have to prove intent. If one is found with one of these agents and one has not registered with the government, that is a violation of the law. When that law was passed, it was also fully recognizing there are legitimate and important reasons why these agents need to be used. And so there's a process by which research institutions and people can become vetted and approved to work with these agents, but it does provide a bar for people who are not within that scheme. Of the gain-of-function pathogens which have been discussed in this context, only two of them, actually 1918 flu and highly pathogenic avian influenza, fall under these regulations. Those are again, this is a pathogen-specific regulation, and those are the two pathogens which have been discussed in this area of, for example, in Griffin's risk-benefit assessment, picking out those specific influenza strains. But I'm sorry, I'm jumping ahead to the DIRT policies. SARS is also a select agent and would be covered by that. MERS is not. So these, again, are pathogen-specific. A third area of legislation which binds everyone in the United States is export controls. These affect the export of certain listed pathogens from the United States or the communication of certain non-public proprietary information, which could, for example, include information on how to develop a particular strain of a pathogen if that were not published in the open literature. Things which are published in the course of fundamental research are not affected by these export controls, but it is a set of statute and regulation that could have some bearing on the ability to do and to disseminate biological research. Then there's a number of policies which are attached to government funding, as we've already discussed. I don't want to say very much about these because I think they're largely familiar. We have the policy for oversight of life sciences dual-use research of concern. This is a mechanism by which the United States government has established a set of procedures by which it will evaluate proposals in terms of does it want to fund them, or are there steps it should take to mitigate risks that the work being proposed may offer. This is also specific to a specific list of agents, which is smaller than the select agent list. And it's this list that only 1918 flu and H5N1 high pathogen avian influenza are in the gain-of-function discussions. The only overlap between that and the existing DIRT policy in terms of federal funding. So gain-of-function research on any other organism would not be covered by this policy. Then there's a more recent policy for the institutional oversight of life sciences dual-use research of concern, triggered by the same list of agents. And whereas the first policy addresses the federal government's decision to fund this work, this policy sets up a process by which institutions doing this work would identify work that needed to be looked at, would establish review procedures inside the institutions, and then would develop risk mitigation plans for particular proposals that required that. A framework which in many respects is very similar to the structure of the NSABB's recommendation is a framework developed by the Department of Health and Human Services for certain H5N1 and, excuse, that's H7N9, not N7N9, influenza, sorry about that. Larry Kerr is going to be discussing that framework in much greater detail, so I won't say much here. And then there's a range of very extensive biosafety guidance, biosafety and biosecurity guidance that the CDC and the NIH have developed for use by anybody doing biological research, and I believe is tied as a term and condition of U.S. funding to be obligatory for institutions receiving funding in terms of a whole process by which research using potentially dangerous pathogens can be done safely for both those working on it in the laboratory as well as for the community. And these two in particular, the BMBL and the recombinant and synthetic DNA guidelines, all address research practices in terms of how to do them appropriately. Again, this is formally tied to government funding, but essentially it constitutes a set of best practices, and I do believe these are used widely around the United States, not for government-funded work, but for all relevant work, and indeed around the world. So even a policy only nominally having the force of any sort of that force is tied only to government funding can have a much greater influence more broadly. And then one other thing I want to talk about, it's actually already been mentioned today. I call them other. It's not federal law. It's not really federal funding guidance. But all research institutions that are working with any potentially hazardous substance, of course, not just restricted to biological organisms, but anything, in fact, not restricted to research institutions, any entity working on something that could pose a risk to its workers or to the neighborhood or to the environment, has to do so in recognition of the fact that should there be an accident, it incurs damages in the community, that they can be held financially liable for that. And so this includes harm not only to the institution's own employees, but it's harm to outside, the general public. And the extent to which an institution could be held liable may depend on the degree to which there is a regulatory structure in place and whether the institution had been complying with those regulations. These can all be used to establish a standard of care and to establish a set of general industry practices, which are all criteria that, should this come to a trial in a lawsuit, may be part of an institution's defense. If it's held to a generally accepted standards or complying with a generally accepted standard of care, that may be a means by which liability would not be incurred. By the way, the big asterisk in my talk here is I'm not a lawyer, so anything specifically I'm saying about liability, you need to check with somebody who actually knows firsthand what they're talking about. But I do want to say the importance of this, because independent of law and independent of regulation, this sets part of the context in which any research with potentially hazardous organisms would have to be judged. And liability also depends on whether a reasonable person, you find a lot of these reasonable people in legal arguments, would conclude that the institution had exercised due diligence in anticipating possible harms and in mitigating against them. So these are among the policy constructs or laws, statutes, regulations, policies that are in place now that any additional development of policy and gain of function would have to be embedded in, and whether these existing procedures would have to be modified to fit the new one or whether they would sit on top would have to be determined as we went ahead. Just to review very quickly, the NSAB proposal is strictly tied directly to federal funding, wouldn't directly affect things outside. That would take legislation. The NSAB proposal does not have a list of specific agents, unlike many of the proposals I've talked about here. As to biosafety and biosecurity requirements, there's no firm specific list of criteria other than, even in the select agent regulations, which do require that institutions have biosafety in mind and have take appropriate care, with language like such as conveyed in the BMBL or such as conveyed in federal guidance. They're in other federal documents. So it's not a specific list of specific requirements, recognizing that biomedical research has a lot of case-by-case determinations that have to be made in terms of what's appropriate in what circumstance. And one other thing I would like to flag, the NSAB's proposal is in terms of the generation, research likely to generate a pathogen described by those three criteria. One question I might put out is, what happens if research to generate such a pathogen has been proposed and has been approved by whatever mechanism we go through? What would govern subsequent use of that same pathogen? Does that have to go through another review, or does that then become sort of like part of the natural background and not subject for any additional investigation? So let me now turn it to my fellow panelists and be happy to discuss these further with you for questions. Thank you.
Thank you very much. It's now my pleasure to welcome Dr. Lawrence Kerr, who is Director of Pandemics and Emerging Threats for the Office of Global Affairs at DHHS.
Thank you, Michelle, and thank you, Jerry, for the introduction. It is indeed an honor to be before you as an ex officio of the NSABB since its inception, and joining you once again from my new position in HHS. It's an honor to be here and to be able to talk about the specific framework that the Department of Health and Human Services has put into effect and has been using for the past couple of years on a very, very specific and narrow set of gain-of-function research, specifically associated with the H5N1 influenza. So we'll take the grand schemes and mechanisms that Jerry described and drill now into the very, very specific framework that HHS has put in place. It's daunting to speak in front of this audience of experts who have been thinking about this for going on 10 plus years now, and it's certainly unnecessary for me to talk about the mechanisms that ultimately have led to the decisions that, as we look at the research continuum from the concept of an idea through the actual execution of that experiment in the lab to the publication, that over time, each of us has recognized that there are inflection points within this continuum where one could potentially address biosecurity concerns. But I think fairly early on in conversations with the NSABB and with the science editors around the world, that clearly the latter points of that research continuum, one finds inflecting at those points to be not only too difficult, too hard, but also too potentially dangerous to the research enterprise to try and put biosecurity measures in place at the point of publication. So if we back up in the entire spectrum to basically pre-award decisions, that is where HHS has really focused, based upon the recommendations of the NSABB, to place its attention on the review process. And in particular, post the 2011-2012 issues associated with H5N1 experiments, the so-called gain-of-function experiments of concern, that is where the particular framework had its origins and the way in which the funding agencies of HHS chose to approach this in a pre-award review manner that ultimately decided upon a departmental level review for certain pathogens within that criteria, and I'll talk about that briefly. So I've talked about within specifically influenza H5N1, the highly pathogenic AV influenza strain of concern, that even within that category, the framework specifically addresses those experiments which are going to go on to potentially create strains that have either increased transmissibility or increased pathogenicity by respiratory droplets. And that's the key, so that if you're taking this world of experiments of a specific pathogen, and now you start to carve off what's going to be done with that experiment, we are talking about a very narrow and defined set of experiments that have the potential to create these organisms through gain-of-function techniques. What one finds is what NIH, CDC, and our other agencies, those are the two major funding agencies, decided was that should these proposals come before them, that the funding agencies do an extra set of level of review where they are specifically asking 7 questions of that particular experimental proposal. These criteria determine whether or not the proposal goes on to a departmental level review that I will describe in just a few minutes. It is important that the proposal, again, around the anticipated production of an H5N1 with increased transmissibility or pathogenicity by respiratory droplets have the following criteria. The virus anticipated to be generated could be produced through evolutionary process. The research addresses a scientific question with high significance to public health. There is no feasible alternative methods to address the same scientific question. Biosafety risks to the laboratory workers and the public can be sufficiently mitigated and managed. Biosecurity risk can be sufficiently mitigated and managed. The research information is anticipated to be broadly shared in order to realize its potential to global health. And the research will be supported through funding mechanisms that facilitate appropriate oversight of the conduct and communication of the research. If the proposal for these specific pathogens meet all of these criteria, then the funding agency refers that proposal to what is referred to the HHG. You'll see why we use the acronym. HHG stands for the Highly Pathogenic Avian Influenza H5N1 Research of Concern Working Group. HHG. I'll use it my entire 5 minutes if I call it that. The HHG is chaired by the Assistant Secretary of Preparedness and Response, Dr. Nikki Lurie. It has on it 12 experts comprising senior officials from HHS encompassing countermeasure development and availability, national security and intelligence, law, domestic and international, public health preparedness and response, select agent regulations, science and public health policy, funding agency representatives, scientific expertise, global health, risk assessment, and ethics. So for every proposal that comes before the HHG, the body is assembled, they review the scientific content that has already been through scientific and technical merit review by the funding agency and put forward to the department. The HHG is then demanded to basically work through a set of criteria to determine whether or not there's basically one of two decisions that come out of it. Either HHS recommends the funding of the proposal or it recommends not funding the proposal. The criteria are that it reviews the funding agency's risk assessments of the potential for the creation of the entity of concern, provide additional and multidisciplinary expertise to consider additional risk factors that may alter the research risk assessment, determine which measures may be needed to mitigate risks, take into account the overall HHS influenza portfolio of HPAI gain-of-function research, and ultimately recommend whether the proposal is acceptable for HHS funding. So in the course of the past couple of years, where, again, this narrowly defined set of research proposals have come forward, only a handful of proposals meeting these criteria have ultimately come before the HHG and have been individually reviewed. And what you find is actually the full spectrum of what you might expect from a review process if it's working well. There were some proposals that received full approval by the working group and were recommended funding to the funding agency director. There were funding proposals that were received, or scientific proposals that were received, in which individual experiments were rejected by the committee and were rejected and were recommended to the funding agency that those not be funded. It also defines why those decisions were made and the potential to mitigate them if they could be addressed by the funding agency with the research investigator. So, in conclusion, I don't want to take up much time. That basically is the overall framework that has been put in place. It has only been used a few times. It is very narrow to the specific subset of H5N1 influenza research that I have described. And with that, I will conclude and take any questions that you have later. Thank you.
Thank you. In a room full of people who can really fling an acronym around, I think you just won the gold medal. Next, I'm pleased to present Dr. Richard Frothingham, who is from Duke University, where he directs the NIAID Regional Biocontainment Lab and chairs the Institutional Biosafety Committee.
Thank you for the invitation. I can be coming from a very different perspective, mostly someone who's been doing this dual-use review as part of an IBC, working with IBC members, working with investigators, and hopefully there'll be some perspectives that'll be helpful from our experience. So we began reviewing dual-use research back in 2003 when the NIH funded the CERCEB, one of the regional centers of excellence, and within the CERCEB they funded a policy, ethics, and law core And one of their mandates was to review the research coming forward for dual use. That was an idea that they had. They thought it was time to begin that process. And they also reached out to IBCs in the region, and I think virtually all of the IBCs from the member institutions came on board and said, oh yes, we'll review dual use. I know Duke and UNC, Emory started reviewing things for dual use, just in general. And they brought some training materials and taught us something about how to do it. There's a publication in Science just describing some of our practical experiences, what worked, what didn't, how the process went, and you can pull that up if you're interested in that experience. For gain of function review by the Duke IBC, we started that a couple years later. In 2005, we added these 7 famous questions about the 7 categories of experiments that could raise concern. And we also added a generic statement of anything else that would pose issues of dual use. We did training for the IBC members in 2006 using the SIRSEB module. And so we've been reviewing protocols, and we do RDNA review. As you can imagine, our IBC also does select agents. We do BSL-3 review, and we review anything that somebody wants us to. So that's how we've gotten these protocols. And so just some of the pathways. I mean, one of the questions is what is the on-ramp, as it were. And in addition to the questions on the IBC form and the concerns that are identified, really IBC members are probably the most likely to identify these. We have gotten queries from NIH study sections and program officers. So that's another process that has come to us, as you can imagine. I want to give you a couple of examples of some gain of function research that we have looked at Duke. I know many of you have examples of how you've dealt with these, and it's an imperfect and complicated system. Every case is a little bit different. The first one that I became aware of was actually one that came through prior to the IBC taking this on, and that was an investigator at Duke who proposed to do some cytokine research with acromegalia. And this is kind of a classic example of gain of function that is problematic, putting cytokines into acromegalia. And this, the dual use concerns were raised by the NIH study section. The investigator had not really dealt with them in the grant application, and the investigator just, I guess, got gun shy and just pulled it back and stopped doing it, just pulled back completely. That's not to say that it couldn't have gone forward if discussion had gone forward, if there was a good justification. We never saw that application, and I can't really speak to why it was justified or not, but that was an experience. And going forward, this has been a sort of a cautionary tale to say, hey, as PIs, we can't be, maybe it's helpful for you to think about these things before going forward. The second one listed there, we see this all the time. All of our bacterial pathogenesis folks are dealing with with virulence factors. They're manipulating bacteria, taking things in, putting things in, taking things out, in hopes of attenuating or making more virulent various bacteria. The reality is most of this research is it's gain of function in a sense. You've got a strain that suddenly produces a colony-forming unit that's twice what the other strain did. But in many cases, these strains, with their souped-up virulence factors, are not fundamentally different from the basic organism. They have the same basic mechanism of causing disease, same transmission, et cetera. In any case, this is an example of one we reviewed it. We said, hey, we don't want you to put 10 different antibiotic markers in the same strain. And if it comes to you from the clinic with 4 resistant to 5 things, you can't put all the antibiotics into the same strain. But other than that, we thought the research was fine, and the PI agreed that if they identified an unusual accentuation of virulence, that they would stop the research and bring it to our attention. That hasn't happened. And we've reviewed a number of these type applications. We got a query from an NIH program officer related to a SIRSEB project that involved adaptation of dengue virus to grow in Drosophila cell lines, taking advantage of genetic tools available in Drosophila to explore how the virus works, I guess. I don't know the details of what they wanted to do with that. And this was actually had been reviewed by the SIRSEB group. They didn't see any dual use to it. But the NIH program officer was reading the 7 things, and it says extending the host range of a pathogen. And if you extend the host range to Drosophila cell culture, sounds like it fits that criteria. The Duke IBC reviewed it. We felt that since it was coming from us from the NIH, that we would like to think about ourselves, but we also recruited some external opinion, external advice on it, and the consensus was uniform that this was not a particularly hazardous approach, because when you adapt a virus to grow in a cell culture, it virtually always loses virulence rather than becomes more virulent, and also Drosophila is not going to become the new host. We just didn't see Drosophila transmitting dengue. So the project proceeded, and we did this typical thing that we always do. We required the research personnel to complete dual-use training, so they're at least familiar with this. And that's actually a step forward, because throughout this period, I would say that most of our PIs, when we ask them a question about dual use, they are not really understanding exactly what we're asking about. They're thinking biosafety usually, they're thinking even biosecurity a little bit, but not so much what is the concept of dual use. A recent one that we reviewed was an HIV infectious molecular clone pseudotyped with VSV-G. And VSV-G is an envelope protein that allows viruses to enter a wide variety of cells. And it's the common workhorse that we use for replication-deficient lentivirus and retrovirus vectors. This is a replication-competent vector. And the researcher wanted to use this to put HIV infectious molecular clones into renal cells to understand how does the renal reservoir work. And this is a one-cycle kind of thing, because the VSV-G is on a different plasmid from the infectious molecular clone, so it's not expected to produce a virus that propagates in that fashion, but the initial infection, the initial organism, the prep, the material would be potentially more hazardous in that it can infect virtually all human cells, including resting cells, including cells that don't express CD4, et cetera. So there was a sense it might be a little bit more hazardous to work with. We actually went for external advice on this one as well, and got some opinions from various people. And in this one particular one, we're beginning to move in areas that you've heard about today. We began to ask the question of, can we work with it safely? What are the risks of this? And then the second question was, is this necessary? Do you need to do this research? And got some opinions on both of those matters. Went forward, the research is going forward, and the IBC felt that it did need to be conducted under BSL-3 conditions, and also with a rigorous administrative controls, and that's ended up with them recommending that it go on within the regional biocontainment laboratory, where there's a lot of infrastructure along those lines. So some of the lessons that we've learned along the ways, and this, I've heard this from people otherwise, some of these things, you've all experienced these as well. So we do encounter gain-of-function studies. These are not the gain-of-function studies that the NSABB is describing or that the HHS framework describes. These are not at that level of heightened concern. And we've not encountered, as it were, gain-of-function studies that are gain-of-function studies of concern. And I've given you a new acronym here, GOFSOC, which I kind of thought was nice. I remember speaking about 6 or 8 years ago when the DIRC abbreviation suddenly appeared. And it appeared in the handout that went to the conference. And I was back there typing away, and I stuck it into my slides. And everyone said, how did you get that in there? Because we just released that. Anyway, I don't know why it's not research of concern. It's studies of concern now. So it's GOFSOC, whatever that means. PIs, in our experience, have had a challenge thinking about dual use. And dual use is so broad and nebulous. All biomedical research has the potential to be misused. This gain of function, and particularly gain of functions being discussed today primarily, is a very well-defined concept. And I think in general, as we talk with our PIs about gain of function studies in the wide range of microbiology, I think it's going to be a little easier for them to understand why we're concerned about this and the potential for misuse kind of questions that arise from it. Consensus is a big issue. And I heard Jerry talk about the reasonable person. And I've got some great IBC members on our committee. And I'd like to think that they're all reasonable. They are independent, they are smart, and they can imagine almost anything. They have great imaginations. So in the realm of dual use potential, what could go wrong, we can think of almost anything. And getting consensus has been a challenge. When I went into this, I said, well, let's come up with some categories, and not dual use, and insignificant, and significant, and I was going to characterize, and I like to characterize things, and that never worked. We could never get consensus on, is this dual use research? Is it dual use research of concern? Is it significant concern, et cetera? All that stuff, we could could never get consensus because we could always imagine some horrible misuse. However, we could get consensus on going forward with risk mitigation strategies, modifications. Those things all happened. So where we weren't able to put people's, we were never able to reach a consensus of this is not a problem, we were able to say, well, let's talk about how we're going to manage this. And management worked across the years. Now, we've never had to deal with the really tough ones, the ones that no matter what we do pose a substantial real risk. That's some of the things we're talking about today. And I'm concerned about the ability of committees to gain consensus, smart people to gain consensus on tough issues. And I don't have an answer for you there. We have often gotten external advice, and I think that's useful when we don't know what to do. It's also useful as a way of creating, eliminating some of the perception of conflict of interest, bringing other people in, getting their opinions so that it's not just all within your shop. How about the benefits? And I've shown you this graphic. We've been keeping tabs on this very closely. These are identified episodes of misuse of Duke research to harm public health, agriculture, plants, animals, environment, or material as coming out of the dual use research of concern. And so these episodes, we're keeping track of them. And prior to our dual use review, we had none that we have identified, and subsequently, similarly, we have had none. It's hard to know if this is working. But I have put at the bottom some things where I think some of the benefits that I think are there. And I hope we never have one, of course. Can we measure benefits? I think public trust in responsible science is maybe the biggest thing that comes out of this, at least at the level of what we're working with, is that people can know that we are reviewing these things, we are thinking about these issues, our PIs are thinking about these issues. Also, early review. People talk about don't review it before the publication. I mean, review it at the very early stages can avoid wasted effort and frustration and maybe even improve the outcomes in the study sections. So I have a few perspectives on the NSABB recommendations. I'm not an expert in this area, and you folks have done so much work on this. I think gain of function is easier to understand than dual use. And in many ways, I'm feeling like we ought to do a more gain of function training our PIs in this concept, as opposed to dual use, which can mean almost anything. The proposed definition of gain of function studies of concern is really very much clearer than our current dual use research of concern definition, which is could be, you know, it says could be directly misapplied, but how is it going to be misapplied? Almost anything you can imagine. This is very precise, and I like that. I think the world of research in this should be small and definable. There probably aren't hundreds of people wanting to do this research. I hope they don't fall through the cracks. I mean, I hope they're all identifiable. Many of them are in institutions that have select agent programs. Many of these are select agents, so I think that expertise will be very helpful. The current IBC or IRE mechanisms, I think entities are going to be important at the institutional level. That's not going to be the only part of the review, but I think that these groups are well equipped. But I think as we go forward, we should have a very low threshold for getting external advice, whether that's from the federal government, whether it's from other experts, just to give us some ability to say we're not just in our own sphere making these decisions. One of the things that we recently did, we've been doing dual-use research within the IBC for now 13 years. We recently moved it to our institutional review entity, the entity we're required to have under the U.S. government policy on institutional review of dual-use research of concern. So we have an institutional review entity. It's a separate committee. It does not have public minutes. We post our IBC minutes on the website. We don't post our IRE minutes. And we believe that dual-use research, it's not always possible for it to be public, because the very fact I'm reviewing something actually might be dual-use information. But it should be transparent. I'm just listing some people here. You can see our IRE members, institutional review entity members on the left side of the right side of the slide, and 3 of them are community members. So we have a community presence on our institutional review entity. You can see the other names there supporting this. Funding for all of this work is institutional. So that's, and we have to pay for this, and it's real expensive, as you can imagine, to do it right. Thank you for, I guess I'm done. Oh, it doesn't progress now that when I get to the end, it won't progress. It throws me out, so I'll have a seat then.
Thank you very much. And our final presenter before we open it up for Q&A is Dr. Philip Potter from St. Jude's Children's Research Hospital. He's a cancer researcher who has participated for many years in institutional governance of dual-use research of concern. Thank you.
Thank you. I'm going to set the stage here for why I'm included. I became the DIRC committee chairman as part of my IBC responsibilities, and DIRC committee reports to the IBC but does not actually publish its minutes, similar to what Richard said, because we have no idea who's reading our minutes and we don't want that science to be generally publicly disclosed. But you might be thinking, why does St. Jude have a DIRC committee? Partly because we have a very large flu program that goes on at St. Jude, run by 2 established PIs. A large amount of money comes through WHO and NIAID. And as part of that, we receive influenza-positive samples from all around the world. The idea is to find out where those viruses are tracking, how they're being spread, what movements are occurring, and are they something we need to worry about? And to do that, we sequence these. They're then categorized based on those sequence, whether they fit into the high path or low path category. And of course, that then covers agents which are potentially DIRC and gain of function. And then based on that, there is biochemical and in vivo studies performed. And frequently, viral segments are swapped around to find out what their function is. So that could, as you can imagine, that could quite clearly fit into the current category of DIRC and potentially gain of function. So how do we go about reviewing this approach? So we set up a DIRC committee, which consists of half a dozen established faculty who have specific expertise in the subject that's being discussed. In our case, it's nearly always flu, of course. We have legal personnel, we have PR folks, we have members from IRB, we have a really diverse group of individuals, and it's up to the PI to espouse how good their science is, risk and benefits, so that everybody from the committee, even from a layperson to a scientist, can understand what the benefits are. And we've been through this process now for 4 or 5 years. We've maybe evaluated somewhere in the order of 14 or 15 different projects. And really what a sort of consensus has occurred is that we've come up with a sort of an internal set of guidelines that we like to use. So I put no gain of function in virus-resistant antiviral agents. That's of course DIRC as well. So I'm using DIRC and gain of function here synonymously. There has to be a vaccine. We typically like to see potential protection of individuals working in that area, so we like the vaccine to be available. We run into difficulty evaluating gray areas, as you might imagine, and the biggest gray area for us arises in the DIRC guidelines, where one of the comments says altering the host range and tropism. So if you take a highly pathogenic virus and you make it less pathogenic, you've altered the host range, the tropism or the host range. Therefore, we have to evaluate that. If it had said increasing, then that would have been okay, but anything that drops in pathogenicity, we also have to evaluate. And we've also mandated that ferret be used as the gold standard for biological testing. That may seem obvious, but some PIs were only doing their experiments in mice to see what the mouse phenotype was, but inadvertently they might be generating something that was also more pathogenic in ferrets, so we just insisted that they do those same studies in a ferret. And to give you an example of something that happened, as was mentioned earlier about H7N9, that framework came in, I guess, probably 2012, 2013, almost exactly at the same time we'd started to see these viruses being detected in samples arriving at St. Jude, and clearly PIs wanted to do that, to do studies on that, but it occurred to us that the studies that might happen would be potentially as hazardous as something being done with H5N1. And so but unfortunately H7N9 is not on the list of 15 agents that is covered by DIRC, so it fell into this hole. We as a committee categorized it as DIRC in lowercase because it wasn't officially, from the government perspective, DIRC, but we realized that those results might just be as enabling as anything else. So, you know, we went back to the investigator and said, you know, these are the concerns. And as a result of that, the investigator decided not to progress because at that time, we knew at that time additional guidance was coming along the line, so we waited for that to be established. So what about the current guidelines that have been provided by the NSABB? I think they provide a really initial good draft, which will certainly help PIs and institutional officials in understanding this. I guess, you know, the problem that everybody has mentioned so far is that this is a continuum, and as a result, there are no specifics. It's all highly likely significant. As somebody who has to sit down and review this information, that's problematic because there are no clear guidelines, no clear milestones for me to say yes or no. So I think the way that DIRC has looked at it is that we've looked at what the starting material is, and then we see whether that would potentially increase that. And so the starting virus is nearly always our sort of baseline. I don't know how else we can do it, but clearly, you know, that we might run into issues at some point. And so the other thing is that, you know, one of the points that's in the document is that, you know, the pathogen generated is likely resistant to control measures. So I think one of the things that St. Jude, the DIRC committee is mandated is that, you know, your parental flu virus that you're going to do these studies on has to be sensitive to oseltamivir before you start doing those experiments. Whether that is a good thing or not, I don't know, but I think it gives us some level of potential safety if anything untoward would happen. So these were the specific questions that Michelle asked me to address, and I hope I've tried to cover them as best I can. So where are the proposals likely to succeed based on what our experience is with DIRC? So I think the criteria are well-founded. I think they cover the right attributes. I think the process that we go through to evaluate this would be pretty straightforward based on our experience with the DIRC committee. I think the expectations for the PI and for the committees to what they have to look for and how they look for it are clear. And I think that our DIRC committee likely has the expertise to assess gain-of-function research. It's not a huge, you know, step here to do that. So where are the major gaps and weaknesses? Now, clearly, DIRC is for the 15 selected organisms, and that included non-avian pathogenic influenza, and that was, of course, the H7N9. So there is still some ambiguity. You know, we fall into this area of where do you start on the continuum and what is good and what is bad? I can't answer that. And I think one of the things that has arisen from our experience is that if the local entity believes something is gain of function or DIRC that doesn't specifically meet the criteria that is mandated by NSABB or other, what do we do in that situation? Does the local entity have control over this science, or is this a government issue that requires backwards and forwards? Does it go through NIH, whoever the funding agency is, to make that decision? I put this up because I think we're going to see more of this as time goes on. And I'd really like the NSABB to at least try and address it. And so what are the most important problems that I think need to be addressed that are, well, I think whatever you decide and however you write it, it has to be crystal clear. It has to be obvious. You can't, if you go through a decision tree like you had previously, you can't have 45 branch points in that decision tree. It's got to be yes, yes, no. Because otherwise, as scientists, we're always looking for that stuff on the edge that doesn't quite fit the hypothesis. You're going to be working around that edge if that's the case. And then I guess the other issue that comes to my mind is that if the local entity has a problem, who do we contact? Is there a group who oversees this, who would be our first point of call when I have to pick up the phone and say, I've got a scientist who wants to do this, who do I need to talk to get a reasoned decision or response? And so again, so my other concerns that weren't listed there, I guess, is, I really think the problem with local description of gain of function, or DIRC, if that system goes forward, you might end up with a patchwork of institution-dependent rules. Now, St. Jude is fairly conservative. I think we tend to be more restrictive on our science than some other places. But I think if you allow local decisions to be made, which I think in some case you might have to, I don't want the system to become even more cloudy than it already is. And so to the last 2 points, I have to state here that I'm a scientist as well as an institutional official. The last 2 comments on me as a scientist don't represent St. Jude's viewpoint. But really, as a scientist, if a PI can really justify to me, DIRC Committee, IBC, the US government, that this is the best experiment ever, is it up to me or anybody else to say, no, you can't do that? It's a philosophical question I think you have to think about, but I don't really want this to stymie the way we do science. And then the last point is really just a validation of that comment. I think we are scientifically more informed based on the work that Yoshi Kawaoka and Ron Fauché did 5-6 years ago about virus genetics. If we didn't have that information, we might not be able to predict what would be coming down the pipeline for viruses that we pull out of nature at the moment. So I think it's really something we have to seriously consider. Thank you.
Thank you. Okay, we have about 35 minutes for questions and answers, and as before, if the speakers would start by identifying yourselves, that'd be helpful. Please.
I'm Pat Fitch. I'm with NSABB, as well as Director of NBAC and President of BNDI. I'm Pat Fitch. I'm an NSABB member. My comments represent my personal perspective, not that of the board. I wanted to start by reinforcing some things that Rich and others said about institutional review entities. We've had a similar experience at our organization where assigning the label DIRC outside the strict government definition became very challenging for the committee. And so as the institutional official for that committee, I switched their charter to instead approve the risk mitigation plan for projects rather than spending their time around whether it defines as DIRC or non-DIRC. And so I think that's likely to become a very broad experience for lots of organizations. The second, I have a question, because I've been wrestling with this myself on the NSABB, and I don't know the answer. I think we talk a lot about the representation in terms of accountability of the sponsor and the performing institution. And so these are organizations that might appreciate what it takes to do the work and the opportunity cost of not doing the work, both scientifically and in terms of the scientific community. But I don't think we've talked explicitly about the accountability of who owns it if something goes terribly wrong, and how they're represented in this process. And so I'd be curious what your perspectives are on how maybe to do that better than we've talked about in the past.
Thank you. One of you want to take that?
So first word is that things haven't gone horribly wrong, which I'm really thankful for so far, and we're doing our best. But one of the things that I've experienced over the years, I have both hats. I'm an IBC co-chair, and then I'm also a scientist. And whenever my research comes forward, I have to leave the room, and they really hold me to this extreme, what I think is a really extreme standard. It took 4 rounds to get my plague work approved, just starting up with anyway, and it's frustrating. However, when news stories would break about a problem here or a problem there, immediately our dean wanted to know what we were doing. And I would generate these memos at 5 in the morning before the news story would get to the dean saying, by the way, you're going to hear about this, and here's what we do at Duke. When the USAA Today report came out, saying all these reports to the, to the NIH Recombinant Advisory Committee, we had reported a lot of things, and we were actually felt pretty good about what we reported and how we'd responded to it. So I think that laying the background very carefully, having the scientists and the review people be independent from each other and holding to a very high standard, it's frustrating, but in the end, you really, when push comes to shove, you're really glad that you have it there.
Let me jump in. Pat, I think there's two answers to your question. One of the reasons I brought up liability is there's going to be a legal answer. If something goes horribly wrong, there will be lawsuits, and the courts will determine that. But there's also a political answer. I think government employees and senior political officials have to have in the back of their mind, what am I going to tell Congress if ultimately the inevitable investigation? So I think that's something that we also have to recognize. And just while I'm here, I want to have an additional word. I'm afraid I may have left a misimpression in terms of select agents and safety. The actual language in the select agent regulations on safety is very small, but the attention given to safety by the program when it registers specific institutions and when it does inspections, and by incorporating the entire BMBL by reference, there's a great deal of attention given to safety. So let me not equate amount of words with degree of attention.
Thank you.
So I have a comment. I think a lot depends on how you construct the committees, and including your IBC, to get as much input as you can at the beginning. So our IBC contains a lawyer who's part of the medical community, that's not affiliated with St. Jude. We also have the public health official for Shelby County, which oversees Memphis. Those people bring a completely different perspective to the way that we look at the protocols, and they raise things that there's no way we would ever contemplate. And I think that's really good. It also happens that the public health official is also our first point of contact if there ever is an exposure at St. Jude, so she can coordinate the exit of those individuals from St. Jude, the safe transfer to an isolation facility in a Memphis hospital. I think having all those pieces in place means that your likelihood of events happening drop significantly. And I think by thinking about those things, which aren't obvious from any of the regulations, but by working through it and thinking about it in advance, you hopefully mitigate those opportunities.
Thank you. Yes, please.
Hi, this is Diane Diolis from National Defense University in the Department of Defense. Formerly at HHS, where I participated in the HPAI review group, which we actually changed the name of that to the HHG because our original acronym was called a bureaucratic troglodyte by Richard Hatchett, so we called it the HHG. My question for the panel, I appreciate all the comments that were made regarding this idea of having the difficulty with crystal clear definitions. And our experiences in the HHG were sort of like a lot of the review of these proposals were sort of on a case-by-case basis, and each one seemed unique. So my question for the group, and for those who've reviewed these kinds of proposals in IBCs or other places, we constantly say that if there is a dual-use gain-of-function study, is there an alternative means for the investigator to do a study that takes them out of the risk range? And I wondered, how much experience do we have with that? Were there any scenarios in which an alternative path of research was recommended, and could that get us closer to helping us define where some of these lines are? Thank you.
So I think from St. Jude's perspective, that's why our DIRT committee has people who are lay individuals. They're much more likely to ask those sorts of questions than scientists are. And actually, I think that's good. We have folks on IRB who ask questions like that, who may not have specific expertise in the science that's actually going on, but they're much more likely to ask the question is, do you need to do that in an animal model? Can you do that computationally? Can you do it this way? which I think is good. Again, it comes down, I think, to making what you think are reasonable choices of having people on committee. Now, of course, when we actually meet as a committee to discuss this, anybody can raise whatever concerns they have. But I think at that point, because the PI has been through this process and will go through this process at quite some length. You know, these are 2-hour meetings sometimes with an hour-long discussion with the PI on the science. They'll have gone through every possible way of trying to do that same experiment without using, you know, a potentially hazardous model to get through. So I think the situation has already been discussed at that point. So I think while that does come up and it is raised, most of the time there isn't an obvious less, you know, what's the word I'm looking for, but there isn't a simpler or more effective or easier way or safer way of doing that.
We've certainly had experiences where we have raised a concern and an investigator has modified their plan, I think more with viral vectors than with these gain-of-function pathogens. But it is, I think it's a tricky balance. Because when you're sitting there as a committee and you say, well, you're working with a pretty safe lentiviral vector, but there's another one that's safer, you know, and you know, do you say to the scientist, you need to go retool with the safer one? That scientist may not have a lot of margin in his research funding. He may be getting pretty close to running out and needing to get his next grant and so forth. And sometimes I think there is a balance to be had there. It turns out that both of them are exceptionally safe. And so we say, well, we'd like you to think about this for the future, but no, we're not going to require you to use the one with the inactivated LTR versus the other. We are going to focus a lot on your Sharps precautions. So it's a balance I think we have to keep so the science can move forward too.
Richard, I was very struck by, I think it was you made an example at one of these symposiums several years ago where you had mentioned something that came up before the IBC and questions were raised about the safety of that approach. And it was suggested to do a different approach. I think it had to do with a neurotoxin, which is virus. Yes. And the answer is that you did a different approach, but you did different science.
Yes.
It was not the same science in a safer way. It was a different approach, and you got different science out of it, which I thought was very telling.
And we actually just asked the question, and the researcher just sort of, I don't like it when researchers just immediately cave and say, oh, okay, we won't do it. And that's not always the right answer. We just want to engage with you and help us understand why it needs to be done. But yes, this was a subbotulinum subunit that is not toxic because it can't get into the cells by itself, but you put it on a retroviral vector and it goes into the cells. So that was the research being pursued. Gain of function.
Greg Koblenz from George Mason University. First off, let me say this has been a very helpful panel and all very interesting presentations. And it ties nicely into a theme that came up this morning in the previous speakers and some of the commenters about the need for learning from past experiences in this domain as we move forward. And I think Duke and CERCEP are to be commended for capturing some of their lessons and publishing them in Science, but that was many years ago. And clearly you have even more experience to draw upon now. And I would suggest both for the HHS methodology, the St. Jude's experience at Duke. I know there are other IBCs that also have taken this on very seriously. A mechanism for capturing these lessons in a more wholesale fashion as opposed to the kind of the retail model we have here. But I think that would go a long way to helping both the PIs understand what are the possibilities, helping policymakers understand what is practical at the laboratory or scientific level, and for helping people understand more broadly the processes behind these risk assessment methodologies. And there was one point that the gentleman from St. Jude's brought up about the risk of a patchwork of institution-dependent frameworks. From my perspective, that's not necessarily a bad thing, because if you have different organizations, institutions that can experiment with different ways of doing these review processes, and they try different approaches and methodologies and different types of members of their committees, they might all learn something different. But if there's a way to share those experiences, right, then everyone benefits from it, as opposed to a cookie-cutter approach where everyone just follows exactly what HHS says to the letter or the select agent, right? Then there's no experimentation, there's no innovation, there's no improvement. So I think there was a value to having different approaches, but there then needs to be a mechanism to kind of gather those differences and have a process of understanding, okay, which of these are actually improvements and do things better, and then some kind of mechanism to incentivize people to then adopt those best practices moving forward. So I realize you guys aren't NSABB, but that would be something to consider for building in how to learn from this process we've gone through, and any future process we establish should have a mechanism built in to provide that kind of feedback so we can all learn and adapt from it.
Would anyone care to take up the question of whether having varying standards from institution to institution is a problem?
I'll chime in and say I actually completely agree with Greg in terms of you're going to have different scale and capacities at different colleges and universities around the states. So the ability to have one cookie cutter model that will fit all of them, I think, is possibly naive. If anything, taking the examples that have been garnered from the grassroots level and that are actually working is what we as a government need to find out, is there a common pattern that exists across all of the models that are working that can be replicated and then molded to the individual university or college's particular frames, customs, the research that they're doing. So I actually think that's a really great suggestion.
I agree with that, with an asterisk. I think it would be useful to have consistency. If one department and institutions is issuing guidelines or doing processes which is clearly incompatible or sync with what someone else is doing, that's not a good situation. But in terms of how one prevents that, I cannot see the government specifying a list of guidelines and specifications and rules and requirements in such detail to prevent that from ever happening. I don't think we could ever come up with that. If there's one thing we know about biology, it's very hard to force it into a box. So I think an inevitable result of the whole field we're in is we're going to have some amount of interpretation and some amount of variability. But rather than mandate consistency, I think the answer is to try and go towards best practices if we can. But here I'm noting the point that one of you gentlemen made about not wanting to release your IRE minutes, because when you're discussing dual use implications of a project, that essentially could also be dual use. So I'm torn between the obvious imperative for transparency, but at the same time, here is a list of all the things we really worry about in our biological portfolio, and having that on a website for everybody to see has a bit of a concern for me. So we have to have some way of identifying and sharing best practices, but doing it so recognizing that we're not just worrying about safety and nature, but we're worrying about people that are mining this to try and do us harm. That's always, at least in the back of my mind.
If I may push this forward just a little bit before we go to the next question, how do we know when an oversight approach is working? Is the absence of a rare catastrophic event in and of itself evidence that we have an effective regulatory framework?
I think it was Richard's point to believe that public trust is essential. And if you have a process which you can lay out and explain and people go, oh, okay, I can see why you're doing that. That sort of makes sense. That's one measure in the absence of did we or did we not have a disaster. So you have to have an approach which one can see why we're doing it. One can see that the things we're doing seem reasonable, and that and basically you have to argue in terms of going through and saying, yes, this is an appropriate response to this issue. If you don't have the ability to collect data, I don't want to get the data on this.
I also think years of reproducibility and then evolution of your process to learn from best practices and challenges along the way starts to define a pretty well-defined program that you can say, at least to date, that nothing has fallen through the cracks. Always with that in the back of your mind that you're on guard for what's around the corner in either research developments or new pathogens of concern that you have to be mindful of. That's part of the evolutionary process.
I think the presence of community members and their stature. I mean, we have the county health director in our committee as well, the medical director of Durham County Health Department, and that's a good place to go for external for community members. One place that I think you can really go a long way with. So I think discussing the details of dual use research is itself dual-use information at some level. It is a problem. There is a lot of dual-use information that is currently published out there, and no one knows it because they don't look at it that way. You start talking about it, and suddenly people realize, oh yes, there is information about that. And I have been to a number, when I go to ASM meetings, sometimes I'll walk up and tap someone on the shoulder and say, you just gave out some dual-use information. We need to not do that. Don't tell me how you do this. Methods for people who need methods, but don't tell the whole world how, whatever it was I told them not to tell them. And I've been to aerobiology conferences where so much is out there. So we need to be careful about dual use information. It's out there, it's published, but it's not as obvious what it is. Where do we get our credibility? One of the places we do it is in the other three pillars that we haven't talked about a lot, which are the biosafety, protecting the worker, the biosecurity, protecting the environment, and the biocontainment, the structure around it. And all of that, it has under an umbrella of administrative controls. And to develop a program that is excellent in those areas, that has the credentialed, by the way, they should be ABSA credentialed individuals out there, or National Registry of Microbiologists, as Joe prefers, but we need to have credentialed people who have the expertise, who are running these programs. We need to have a structure that we can point to and say, yes, here's how we protect people, and focus on the biosafety, the biosecurity, and the biocontainment, because those are things that we can talk about a lot more freely.
Thank you.
Could I just chime in and say kudos to you for including a public health person, because that's a person that's often forgotten about in an IRE. The second I often get a question of, how does an institution bring a security person on board who actually understands the world of the security community? Certain IREs have brought in their local WMD coordinator, who is a person who is trained in the security and trained in the threat and risk assessment environment to be able to lend that perspective, sometimes from an outside perspective to an institution. But those are often disciplines that we'd like to see incorporated into that.
Our WMD director, our local director, provided our initial insider threat awareness, and clearly insider threat is what the real threat is. We all know that.
Sorry, although not part of our IBC, we have an FBI contact who comes regularly. Yeah. We discuss with these people, so that they're aware of what's going on. They probably don't know the nitty-gritty because they don't need to know that, but they are aware of our security facilities and how we coordinate them.
Mm-hmm.
Thanks for your patience.
Yeah, thank you. Silja Bönicki, University of Freiburg, and German Ethics Council, Harvard Law School Fellow at the moment. I have a question to Gerald Epstein, because I was very glad that you mentioned the liability issue. Mm-hmm. And my question would be whether you thought about a duty to be insured for research organizations, because if we would have a duty to be insured against accidents, there would be another element of cost-benefit ratio into this whole topic, I think. This would be my first question. And the second question to Gerald Epstein as well. You talked about the bioweapons and chemical weapons treaties, and my question would be whether your department or the government thinks about an initiative to either enhance these treaties in regard to gain-of-function research, dual-use questions, or whether there should be another international treaty.
Let me duck the second question. There's an active interagency program within the United States government looking at how these treaties should be strengthened, and lots of thoughts are given to various ways in which that might be done, but I won't have a specific answer as to that particular proposal here. On the duty to be insured, at least my department does require laboratories doing work with facility agents to have insurance, and that is one mechanism by which one can try and influence behavior of institutions. The problem with any insurance approach to particularly a security problem is being able to come up with an actuarial calculation that if you do these things, then you will save money because bad things won't happen. It's very tough to make that connection, and it's very hard for an insurance industry to then issue rates that are commensurate with the risk. But certainly from a safety point of view, I think that is a tool that is being used, that institutions facing liability threats would need to procure insurance, and then whatever requirements the insurance companies would place on that will modify the behavior or reinforce good behavior by those institutions.
Okay. Dr. Bloom.
Comment and a question to particularly to Jerry Epstein. In the beginning, you began with the 3 criteria, and I am really now quite puzzled by the third of those criteria. The original criterion, number 3, in addition to transmissibility and virulence, I'm Barry Bloom from Harvard School of Public Health, was that it be resistant to existing control measures. And I was offended by that because while we may have Tamiflu in this country and vaccines for flu in Bangladesh or in Burkina Faso, they don't have the capacity to control things, and I thought that introduced a level of inequity. The current formulation, as I gathered it from this morning, is that it have an increase in virulence, transmissibility, or ability to spread in populations, which to me is the exact same as transmissibility. Why add a third criterion when it's already redundant with transmissibility? And I'd be grateful for you and the NSAB in general to clarify that. And the fewer restrictions and the clearer they are, it seems to me the better it is. My question to the panel is, which I thought was a wonderful panel, is there a need for separate institutional panels or even higher level panels for GOF and DIRC? Would it make sense to consolidate those activities and create a group of experts since they seem to overlap?
So we don't have a gain of function panel as yet. The history of the gain of function of St. Jude is that we had a big grant submitted. It was funded. It was flagged by NIAID as having potential gain of function science in. It was sent to St. Jude and said, you need to evaluate the gain of function in this. So we convened a committee of 15 or so senior faculty, senior administrative folks, IBC personnel, security individuals, and we evaluated the science, and we gave everybody a score sheet, and there were maybe 10 or so projects that were up, and the question was, do you think this was gain of function? And there was a whole range of 100% yes to 100% no to everything in between. And clearly we realized that, this was never going to be an obvious, easy decision. We sent all that information to NIH. All the non-gain of function came back and said, yes, you're happy to continue that. We as an institution decided not to continue with any of the gain of function because, it was hard to know exactly what the criteria was at that moment, so we were still under the pause, so all that science has essentially been stopped. Ultimately, I think what will happen is that gain of function will be reviewed by our DIRC committee. We may well add a couple more individuals who have expertise, potentially in biosecurity, biocontainment, to maybe fit with that. But it's still a work in progress, but I'm pretty sure that we have the right expertise at the moment. We just might need to expand it to cover that, separation.
Let me take your first point. I now have the opportunity to duck two questions in a row. As an ex officio member, I was relaying the NSAID working group's point, but I'm not the one to answer the question. However, they are in the room here, and they heard your intervention, so I think you have had the effect of speaking to the working group with that.
Thank you.
Okay. Thank you. Yes, please.
Hi, Allison Mystery with Griffin Scientific, and Jerry, my question is for you as well. So I appreciated the policy landscape that you laid out for us, but I'm curious about your statement that the institutional DIRC policies or the federal DIRC policies don't cover this type of research. The summary for both policies states that the list of organisms will be reviewed and expanded as necessary, so I'm wondering why we wouldn't update those policies instead of and update those policies as needed rather than create an entirely new policy that institutions needed to react to, especially when they've been expected to set up these new systems, the IRE that the two institutional representatives mentioned. And also that policy, the scope is meant to address people who both fund and conduct research. So the scope is, as far as the institutions, a little bit broader rather than just the funders. So I'm wondering if you could speak to why we wouldn't just expand or repurpose that policy instead of creating a potentially duplicative one with potentially a smaller scope.
Sure. What I meant by not covered is as they are now, and that's largely because they're based on a list of 15, 16, 17 specific agents, and so if you weren't working, and that's the first test that one applies to see if those policies are relevant. So if one isn't doing one of those listed agents, then one isn't even in the discussion. One could well imagine expanding those policies in some way to capture this, and I think a lot of what I've heard earlier was a request to try and have an integrated approach and not have a lot of eaches and everys. Let me say one way, though, in which there's a kind of different in focus, and I'll throw the select agent regulations as well. The DIRC policies and the select agent rules, which are predicated on listed agents, are predicated on what you start with. Gain of function is all about what you end up with. So for example, the select agent rules, if you take a non-select agent and transform it into an agent that would be as scary or as relevant to the security community as a select agent, it doesn't make it a select agent. So the policies that are triggered by a list of what you start with really, unless they're modified in some more general way, aren't able to accommodate things that might be outside your list, but by the time you're done working on it, ends up being something that would have been in your list if it had existed initially. So it's a sort of a philosophical approach in how those are done, and I think that would have to get reconciled.
But just to remind you, the second screen is to cover the 7 experiments of concern, which do cover the gain-of-function phenotypes.
But you never get there if it doesn't pass the first screen, the way the policy is not configured.
That's true, but the policy is explicit in telling institutions that they are not limited to those 15, that if they are concerned about any organism, they are invited to apply this same methodology to those. It's not limiting the institution at all.
No, it doesn't limit institutions, but and we certainly do encourage that. But in terms of what the government has an expectation to require for recipients of funds, we can't hook that extra piece on.
So I could comment a little bit on that sort of the interrelationship of those, because as I mentioned, Duke's been reviewing every research protocol for dual use, regardless of what pathogen it is or anything. So we have covered the whole water all the way through. We certainly do not intend to start putting all of our dual use review that we do routinely for 10 applications a month into the U.S. government policy on institutional review of dual use research of concern pathway. That pathway is a laborious pathway. If we need to go, and if we need to get help, we'll get help. But there's a lot that goes through that doesn't go down that pathway. And I guess I can't picture a lot of institutions voluntarily saying, hey, I've got some Shigella research going on here that's kind of concerning me. Maybe I should go through this pathway and call up the program officer and say, well, I made it through the first and the second, and now I need to notify you that. I just don't see that happening. I think the policy, it's a list-based, select agents are list-based, and the U.S. government policy are list-based, and nobody adds to these lists unless they're forced to do so. One question that comes up, though, is, and this is where the gain of function comes in, how broad should this be? We've mentioned 3 or 4 pathogens. Are there others? And I was also struck that the gain of function framework recommendations from the NSABB is limited to human health. And I wonder about animal and plant issues as well. Certainly the select agent world covers a lot more than just human health. Just ideas out there.
Thank you. Yes, sir.
Hi, Chris Park from the Department of State. Actually, just on this last issue, to jump in on something I wasn't actually intending to raise, but there's one other, I think, important distinction, at least at the moment, between the gain of function and DIRT categories, and that's what gave rise to the funding pause, et cetera. It was not just the concern that you were potentially producing enabling information that could be misused, which is the underlying concern that drives the concept of dual use research of concern. It was that we may have underestimated the safety risks. That was, in fact, a central focus of many of those who were expressing concern. It was clearly one of the elements that went into the decision. Doesn't mean you couldn't develop a single tool that addressed both, but there are some different issues going on in this discussion. And I think when you're trying to figure a solution out, it's really important to keep in mind, what am I trying to fix? Because you might actually need a different tool. But actually what I wanted to comment on was I was struck that, again, and every time there's a meeting on this topic, there's a lot of focus on, well, we need clarity, we need clear, crisp definitions, et cetera. Mm-hmm. And yet, what struck me was I listened to Dr. Kerr and I listened to Dr. Frothingham, and your descriptions of your processes sounded actually very useful, very overall successful. And it occurred to me, you were both in situations where you don't really have that problem. In Larry's case, the boundaries for what goes into that review process are pretty straightforward, at least as I understand it. Plus, it's somebody else's job to decide it needs to come to you guys for review, which is always nice. And Duke has a very broad sort of ecumenical approach in terms of everything gets reviewed. So the focus was on, well, what's the problem here? What are the benefits? What are the alternatives? Which, from my perspective, is where I want the brain power going, not into a definitional exercise of does it fit in this box or doesn't it. And at the same time, we hear, on the one hand, we need this clarity. On the other, the places where we've tried that, say the DIRC policy, the first thing that happens is people start pointing out, well, that fell outside, there are gaps. So I worry that if we are trying to produce clarity in terms of creating a category where you say, does this require review, then there's a real risk that you wind up in the effort to achieve both clarity and a small manageable category. You either result in considerable ambiguity, which people find frustrating, is uneven, et cetera, or you wind up with something that's got some pretty obvious holes. So the question is whether it's maybe better to have a broader basket of stuff that gets reviewed with the understanding that, well, what you do with it and how long it takes you to review it is going to vary very much depending on some fairly practical considerations. Obviously, if your category, if there are big consequences attached to a category, if it falls into, basket X, it means it shall not be done. That's a different story, but I'm not at all persuaded that's where we're headed. So just a thought that maybe those two things, maybe sometimes we put too much effort on the definitional question rather than on what can be done about it. The related question is, I was also struck by a couple of the examples of things that have been done, how people have identified issues and what has been done. That's where I don't think we have a big enough basket of examples of tools. And at the same time, we're now recognizing the fact that review, the considerations itself are potentially dual use, so we're taking that information offline, doing it in settings without minutes. Makes sense, but it seems to me we really need to find a way to, without putting it on Facebook, share that information amongst review entities and the government to start developing clear models. I think one of the things that will take a lot of the sting out of this whole exercise is the extent to which people can show successful ways of identifying, prioritizing, evaluating, and particularly addressing risks when they're identified. If we can do that, the whole process is going to get a lot less painful because people will say, I've seen a couple examples, I got an idea how to deal with this. Whereas right now, everybody seems to be doing it de novo.
Thank you. Well, there's a lot in there. Everyone want to dive in?
I think it's impossible to categorize the wide range of biological research that can be done. And I think even in the very limited area of influenza, it's not going to be possible to, this 500-page document, wow, I'm really impressed. And I'm sure that tomorrow someone will think of an experiment that's not covered in it. That's what science is about. It's always going to be, every case is going to be a little bit different. It's also what makes life interesting.
I think my concern is that the minute you start going down the dual use and likely gain of function, if you do meet the second criteria, you have to then contact NIH. Yeah. You then have to get approval. The complexity grows significantly. What I don't want to happen is in gain of function is that the first two criteria are so sloppy that you're contacting NIH every time you want to do an experiment. I think that really becomes an issue, and that's what I want to avoid.
Chris, I would contend you're right that whenever we're dealing with a very narrow scope, it actually is much easier to define the framework. Mm-hmm. The thing that I would note from the previous presentations is the power of having the discussion. I think one of the powers that you started over a decade ago, sensitizing your community, your staff, to having the conversation so that when you come across something in 2015, that board can then take something new and can actually have a robust discussion on, does this meet your definition of dual use research of concern? But more importantly, what do you do about that? And to me, that's actually the more important part of the conversation for not only the university and the PIs and the laboratory workers' safety and security, but for, as you said, the peace of mind of the community members and their fidelity or respect for the fidelity of your process.
I'm very struck by Dr. Frotherham's remarks, and also corroborated by Pat Fitch, that it can be a great deal of effort which is not necessarily useful to decide whether a definition fits when that effort is much more readily applied to what one does about it. On the other hand, if one has a set of experiments which are designated as needing review because there are particular concerns, particular risks, we are defining a category where we have to apply separate scrutiny. So we are differentiating that set from everything else. So I would like to have a continuum where we don't have to bother with arbitrary definitions. Let's just talk about what we need to do. But we do recognize that there is a certain area of work where we need to do more than we need to do for everything else. And we don't want to make that consistent by having that level of scrutiny for every single thing we do. So we want to differentiate the scrutiny given to things depending on the risk they pose. And hopefully without having a lot of definitions about is one in a box or not when that is irrelevant. But recognizing one does have to make a distinction somewhere along the way. And so how do we help reviewers decide what needs that scrutiny and what doesn't?
Thank you. Well, I'm going to adjourn this discussion for now as we reach the end of our session, but we'll be picking up our discussion of policy approaches back up at 3:45 after the break. Would you please join me in thanking our excellent panelists?